What if the healthiest foods in your fridge are quietly damaging your cells? Dr. Georgia Ede reviewed dozens of controlled human trials, and the results shocked the nutrition world. Vegetables, long praised as disease-fighting superfoods, may actually be fueling inflammation, leaky gut, and metabolic damage. Let's uncover what's really happening inside your body. Your vegetables are slowly poisoning your cells. Dr. Georgia Ede reviewed 24 controlled human trials on whole foods, fruits, and vegetables. The results contradicted decades of nutritional orthodoxy. Vegetables showed limited health benefits. In some trials, they produced slightly harmful effects compared to fruits. This finding challenges the fundamental assumption that plant foods are inherently beneficial for human health. The evidence suggests something more complex is happening at the cellular level. The epidemiological illusion. Population studies consistently link higher fruit and vegetable intake with better health outcomes. These correlations have shaped dietary guidelines for generations. The problem lies in what these studies actually measure. People who eat more vegetables tend to exercise regularly, avoid smoking, and maintain higher socioeconomic status. These confounding factors make it nearly impossible to isolate the true effect of plant foods themselves. This phenomenon is known as healthy user bias. It creates the appearance that vegetables cause better health when the relationship may be coincidental. Controlled trials that remove these confounders tell a different story. When vegetables are tested directly against other dietary interventions, their benefits often disappear or reverse. There's chemical warfare in your salad, okay? Plants cannot run from predators. They evolved sophisticated chemical defense systems instead. The Toxic Plants Phytotoxins database catalogs 1,586 phytotoxins from 844 plant species. Scientists estimate at least 20,000 different plant toxins exist, ranging from mild irritants to lethal poisons. These compounds serve a specific evolutionary purpose. They deter consumption by making the plant unpalatable or harmful to eat. Modern edible plants still contain these defense chemicals. Domestication reduced their concentration, but did not eliminate them. Vegetables contain higher levels of these compounds than fruits. Oxalates bind calcium and form inflammatory crystals. Lectins damage gut lining. Tannins interfere with mineral absorption. Glycoalkaloids in nightshades can reach toxic levels. Humans lack the specialized digestive systems that allow ruminants or hindgut fermenters to break down these toxins effectively. Many pass through the human digestive tract largely intact, potentially causing systematic inflammation. What happens inside your cells? Well, oxalates provide a clear example of how plant compounds cause metabolic dysfunction at the cellular level. When consumed, oxalates bind strongly to calcium ions. This creates insoluble calcium oxalate crystals. Approximately 80% of kidney stones are composed of these crystals. A normal portion of spinach contains 500 to 1,000 milligrams of oxalate. This significantly increases urinary oxalate excretion and stone formation risk. The damage extends beyond the kidneys. Calcium oxalate crystals physically damage cell membranes and organelles, including mitochondria. This disrupts ATP production, the fundamental energy currency of cells. The immune system attempts to contain these crystals by activating inflammatory pathways. Persistent oxalate presence causes chronic immune activation, leading to tissue damage and autoimmune responses. The chronic inflammation contributes directly to insulin resistance and metabolic syndrome. Lectins operate through a different mechanism but produce similar systematic consequences. These proteins bind to carbohydrate molecules on gut epithelial cells, damaging tight junctions. This increases intestinal permeability, allowing toxins, bacteria, and undigested food particles to enter the bloodstream. Research shows 100% of tested patients were positive for leaky gut. Resolution occurred in 91% following a lectin-free diet. The resulting chronic immune activation can trigger autoimmune diseases through molecular mimicry. Lectins mimic human cell surface molecules, causing the immune system to attack the body's own tissues. The broccoli paradox. Nutrition science celebrates broccoli for its sulforaphane content. This compound activates the NRF2 pathway, increasing production of endogenous antioxidants like glutathione. This sounds beneficial. The mechanism tells a different story. Sulforaphane does not act as a direct antioxidant. It functions as a pro-oxidant, causing mild oxidative stress and cellular injury. The NRF2 activation is the body's emergency response to this damage. Cells are going into detoxification mode, not receiving protective benefits. Research confirms sulforaphane causes DNA double-strand breaks in healthy cells. It also acts as a goitrogen, interfering with thyroid function by competing with iodine uptake. Broccoli stores sulforaphane precursors separately within its cells. When the plant is chewed or damaged, enzymes mix these precursors to form the toxic compound as a chemical defense. Consuming broccoli activates a natural plant toxin designed to deter predators. The body responds as it would to any toxin by attempting to detoxify and repair the damage. Nutrition science isolates sulforaphane, celebrates the antioxidant response, and misses the underlying cellular injury. This reductionist approach obscures the complex biochemical reality of whole food matrices. We bred poison into food. Wild plants are largely inedible. Their natural form contains toxin levels that would sicken or kill humans. Over thousands of years, selective breeding reduced bitterness, toxicity, and inedible traits. Modern edible plants are dramatically different from their wild ancestors. Cucumbers once had toxic compounds and protective spikes. Carrots were bitter, pale roots. Avocados had huge seeds and minimal flesh. Bananas were bitter and full of seeds. Corn evolved from a grass-like plant called teosintine. The domestication process was essentially detoxification, right? But humans engineer plants to be tolerable, not inherently healthy. Even domesticated plants still contain defense compounds at reduced levels. They require further processing through cooking, soaking, or fermenting to minimize harm. The notion that plants are naturally health-promoting ignores this history. The original forms were weapons against consumption. Modern versions are simply less poisonous. And then there's the fiber myth. Fiber is promoted as essential for gut health and metabolic function. The cellular reality contradicts this narrative. Fiber is indigestible plant material that humans cannot break down or absorb. It provides zero nutritional value. Insoluble fiber causes microabrasions and irritation to the gut mucosa. This triggers increased mucus secretion as a protective response. This mucus production is often interpreted as a sign of gut health. It actually indicates irritation and inflammation. Soluble fiber undergoes fermentation by gut bacteria, producing short-chain fatty acids like butyrate. These compounds do support colonocyte function and have anti-inflammatory properties. However, similar benefits can be achieved through ketone production on the low-carbohydrate diets. Beta-hydroxybutyrate, a ketone body, functionally overlaps with butyrate in fueling colonocytes. This endogenous ketone production bypasses the need for fiber fermentation entirely. It avoids the gut irritation, nutrient binding, and potential dysbiosis that excessive fiber can cause. Improvements in biomarkers associated with high fiber intake often reflect displacement of refined carbohydrates and ultra-processed foods. The benefits come from removing harmful foods, not from fiber itself. what actually optimizes metabolic health. An evidence-based approach to metabolic optimization prioritizes reducing exposure to plant defense compounds while supporting cellular function. The most critical first step involves gradually reducing oxalate intake. High oxalate foods include spinach, chard, beet greens, and certain nuts. Rapid elimination can cause oxalate dumping syndrome, so gradual reduction is essential. Next, address lectins through proper food preparation. Soaking, sprouting, and pressure cooking significantly reduce lectin content in legumes and grains. Of course, not eating legumes and grains reduces that completely. Finally, consider moderating fiber intake to allow gut healing, particularly for individuals with inflammatory bowel conditions or a leaky gut. The dietary framework that emerges emphasizes animal-based foods as primary sources. Meat, fish, eggs, and animal fats provide essential nutrients without plant anti-nutrients. The foods support gut barrier integrity through amino acids like glycine and threonine. They enable ketone production through dietary fat, which fuels colonocytes without requiring fiber fermentation. Early improvements typically appear within 2 to 8 weeks. Joint pain decreases, brain fog lifts, gut symptoms improve, energy increases. Metabolic markers shift over 3 to 12 months. Insulin sensitivity improves, lipid profiles stabilize, and inflammation markers decrease. Long-term outcomes include disease remission, particularly for autoimmune conditions. Cardiovascular health improves, body composition optimizes. So if we got all this, why does this evidence stay buried? This perspective fundamentally challenges the plant-based nutrition framework that dominates public health messaging. Multiple barriers prevent mainstream acceptance. Institutional inertia plays a significant role. Medical and nutrition fields rely on established guidelines developed over decades. Admitting these recommendations might be flawed requires paradigm shifts that institutions resist to maintain credibility. Economic interests, i.e., follow the money, create powerful resistance. Food industry stakeholders producing plant-based products have vested financial interests in maintaining current guidelines. Research funding often comes from these industries, creating conflicts of interest that bias outcomes. Ideological commitment intensifies resistance. Plant-based nutrition is intertwined with ethical, environmental, and social justice movements. This makes it more than a dietary choice. It becomes a cultural identity. Advocacy groups frame opposing evidences as threats to broader social goals. Scientific complexity provides cover for maintaining the status quo despite contradictory mechanistic evidence. The combination of industrial conservatism, economic incentives, ideological commitment, and scientific uncertainty creates an environment where paradigm shifts occur slowly. Alternative perspectives struggle to gain traction even when cellular and biochemical evidence is compelling. The individual calculation. The harm-benefit calculation for plant compounds varies significantly by individual. Genetic differences, gut microbiome composition, and baseline health status all influence tolerance. For most people, consuming standard Western diets, adding vegetables initially improves health markers. This improvement primarily reflects displacement of ultra-processed foods high in refined sugars, seed oils, and artificial additives, none of which you should be eating in the first place. The body may also respond to mild stress from plant toxins with adaptive hormetic responses. These temporarily boost antioxidant defenses and metabolic function. Over time, chronic exposure to plant toxins can overwhelm these defenses. Inflammation accumulates, gut integrity deteriorates, nutrient absorption decreases. The tipping point where harm outweighs benefits varies by person. It arrives sooner for those with pre-existing gut issues, autoimmune conditions, or metabolic diseases. Understanding these cellular mechanisms provides a framework for individual optimization. It challenges the universal recommendation to eat more plants by revealing the complex biochemical reality beneath population-level correlations. The evidence suggests we are eating less poisonous versions of plants, not inherently healthy foods. This reframing opens space for dietary approaches that prioritize cellular function over epidemiological associations. Metabolic health optimization may require reducing, not increasing, exposure to plant defense compounds. Individual experimentation within this framework, guided by objective biomarkers and subjective well-being, offers a path beyond current dietary orthodoxy. Okay, thanks for watching, everyone. If you have any questions or comments, please put them in the appropriate section. Let me know. We'll answer every comment within reason that we can. And also, if you haven't done so, please like, subscribe, and share this video with someone else. It helps the channel a great deal, and we thank you. You're going to be helping a bunch of other people. Thanks.